What is it that you've committed your life to doing, Thomas?
Well, we're right now committed our lives to managing cancer effectively without toxicity, which is based on the science that I and others have done in this field.
You have a perspective on treating cancer and other metabolic diseases that others don't have. The mainstream, should I say?
Oh, yeah. Well, mainstream doesn't have it for sure. But it's based on science. I mean, my work is based on what Otto Warburg, the famous German scientist, said from the 1920s, 30s, and 40s. He clearly showed that cancer was a mitochondrial metabolic disease.
What does that mean, mitochondrial metabolic disease?
Okay. It means that the origin of the disease resides in the organelle called the mitochondrion. It's in the cytoplasm of the cell. It used to be called, and still is, the powerhouse of the cell. It gives the cell the energy.
I think we have a mitochondria. Could one of my team bring in a mitochondria?
Well, you have a mitochondrion here. Oh, here we go.
We've got one each here.
Yeah, well, see, this is the little organelle that you see. It looks like a bean shape, but it's actually a tubular network. These are tubes, and they respond dynamically inside the cell to both internal activities as well as external activities. So you have to realize that at the time of conception, all of the mitochondria for the developing embryo are in the cytoplasm from the mother.
The cytoplasm.
The mitochondria are not in the nucleus; they're in the cytosol.
Oh, outside of it.
Outside of the nucleus, but in the cell body itself. So all of the mitochondria, they determine our destiny. They will determine how long you will live on the planet if you don't have an unfortunate accident or something like this. They have an expiration date. Different species die at different times. You don't find people living 400 years. Mice live about 2.5 years. Elephants live, as long as we do or whatever. But that's all determined by this organelle. So you can see I have wrinkles and this kind of thing. This is from living on the planet. And this is from wear and tear on this organelle, which allows us to make energy efficiently. Mm-hmm. So when this organelle starts to falter with age, you die. You die from old age. This organelle has to be protected and respected if you would like to live a normal lifespan. But in diets and lifestyles and the way we are today, we damage this organelle. And this organelle then can present itself damage to this organelle, which is a tubular network inside the cell. But let me say something else. It not only controls the internal environment of the cell, it also controls the neighboring cells, the liver neighborhood, the lung neighborhood, the colon neighborhood, the brain neighborhood, the glial neighborhood, the neuron neighborhood, but they all come from the same origin in that cytoplasm. And they determine the overall metabolic health of your body. It's a systemic. They communicate with each other across cells, across tissues. I'll tell you, this organelle controls a lot of what that nucleus does. It tells the cell when to divide. It tells the cell when to slow down. It's
kind of like a brain, but also like an engine room.
It's kind of like that. Certainly, the brain part of it is really mysterious in the sense of how it controls the destiny of the cell in the body.
So sickness, disease, cancer. What do we know about the role that this little thing plays in these chronic diseases and illnesses and cancers that so many people suffer with?
Yeah, well, this is the organelle that becomes damaged. And it can be damaged in many, many different ways. For cancer is what we have spent a lot of our time on. And now we've moved into the whole chronic disease issue because each chronic disease can have different manifestations of ill health to the mitochondria in a particular population of cells. But in the case of cancer, which is what we call the most serious of the chronic diseases, creating the most trauma, the most emotional distress. But we have clearly shown, based on many, many works, that multiple things from our environment can damage this organelle in a particular population of cells in a particular organ. For example, when you talk about carcinogens, it's a chemical that causes cancer. How does that chemical cause cancer? It damages the proteins and the lipids. These little squiggles are delicate internal membranes. They contain the proteins and the lipids that allows us to generate energy when we breathe. Okay, you're breathing, I'm breathing. I take in oxygen. Oxygen serves as a final acceptor for electrons that allows ATP, to come out of this little, and don't forget, it's a tubular network.
And ATP is the energy currency.
It's the chemical energy currency. It allows enzymes to work. It allows all the metabolic machinery inside of a cell to work optimally.
So just to play this back to you so I know I'm clear, oxygen comes in because I breathe in, I then eat food. In that mitochondria, it does a process and it spits out ATP as the energy.
Well, and the waste products of a good energy metabolism would be CO2 and water. So when we burn gasoline in an engine of a car, we break down the octane, the carbon-hydrogen bonds in an octane, and we have an internal explosion that drives pistons. Okay, the exhaust is a lot of waste products from breaking down. We're doing the same thing inside the cell. We're combusting carbon-hydrogen bonds. And that combustion of carbon-hydrogen bonds is a graded process. So it's not an immediate explosion. You're breaking down the carbon-hydrogen bonds in a very precise way, producing ATP, which then drives the entire machinery of the neurons and the rest of the body. Mm-hmm. It can respond dramatically to energetic stress, emotional stress. Anyway, I gave you an example of a carcinogen. Intermittent hypoxia, like people who have sleep apnea, they stop breathing for 30 seconds or more in that general. And then that creates ROS. And that's what carcinogens do, ROS. These are called reactive oxygen species. They damage those delicate membranes. And then what happens if it's too acute, too stressful, the whole cell will die. The cell loses its energy and we get apoptosis or necrosis, cell death. But if it's gradual, chronic, over years, months, years, this organelle loses its ability to produce sufficient energy, but the cell compensates, interestingly enough, by using these ancient pathways, heirlooms of our evolutionary past. So, because all life on the planet evolved in oxygen, without oxygen in the dark, these cells grew like crazy. They were single cells, they were unbridled proliferation and all this kind of stuff. They didn't have mitochondria. They had bacteria. And the bacteria, which this organelle came from, was a fusion between one cell that had a nucleus and was fermenting through the cytoplasm. And this bacteria, which is the mitochondria, came in. And now you have two different forms of energy. You have the energy in the cytoplasm, the ancient fermentation, and then you have this new form which can take in oxygen and generate energy much, much more efficiently than the heirloomic. Listen, one of our big discoveries, if you can believe it. But you see that space in the middle there?
Yeah.
That's called the matrix. And that's where the Krebs cycle, the TCA cycle, which breaks down the food that we eat. But they have an ancient part of a fermentation mechanism inside, because before oxygen came, all life forms were fermenters. They produced energy without oxygen because there was no oxygen. We had to wait for those bacteria to make oxygen through a photosynthetic process. But all organisms were fermenters in the beginning. After this organelle came in and was able to take in oxygen, make energy really, really quick. But in that matrix they have, in the cycle, there's a little pathway there that makes energy without oxygen from the evolutionary past. So we have it in the cytoplasm of the cell because they can ferment in the cytoplasm. But this organ, our big discovery, with the work of Christos Chrounopoulos from Semmelweis University, he's the world leader on that little pathway. When this organelle becomes impaired, these ancient pathways of energy through fermentation arise. Okay, they try to replace the lost energy from the efficiency of this organelle. That space starts throwing out ATP from glutamine. It's another fermentation fuel, and it's making ancient energy in the sophisticated organelle that evolved to make efficient energy. So let me tell you. So you asked me, what about damaging this organelle and what happens if the damage to oxidative phosphorylation?
What does that mean?
Which means energy through oxygen, is too acute, the cell will die. Cyanide is a perfect example of this. You take a mouse or a rat or a person and you drink Kool-Aid, the cyanide-laced Kool-Aid, you die. Because what happens is that cyanide binds to the protein that's going to use oxygen for energy. And the whole system shuts down.
Like suffocates you, basically.
Yeah, you die instantly. And there are other things that can kill quickly, azide and a variety of other chemicals. But for chronic diseases, it's usually not an acute stress on this organelle. It's a chronic stress. And in cancer, what happens in a particular tissue, whether it's bone, lung, bladder, brain, they gradually compensate with these ancient fermentation pathways. So this organelle signals to the nucleus, I'm suffocating, I'm not getting enough energy. So the nucleus turns on the transporters on the surface of the cell to bring in fuels that will elevate energy through what we call oxygen-independent mechanisms. This is an oxygen-dependent organelle. This gets most of its energy because we breathe. All of our cells are using oxygen. And the CO2 that we're blowing out and the water that will be collected in the form of urine when you might put other waste products in there, that's efficient metabolic homeostasis. This organelle makes not only the cell, but the whole body in a state of metabolic homeostasis, where all systems are working at optimal efficiency. But with chronic disruption, smoking, lack of exercise, you can go right down the list of all of the different things that can elicit cancer. Any kind of carcinogens, microplastics, forever chemicals, glyphosate, and any of these kinds of things that would chronically damage the ability of this organelle to produce energy. Viruses, oncogenic viruses, inflammation. You have chronic inflammation. Any of those things damage the sophisticated ability of this organelle to produce sufficient energy. That's why the oncogenic paradox, which we solved.
What's the oncogenic paradox?
That was the paradox that was first put out by Albert Szent-Györgyi, a Hungarian scientist who received the Nobel Prize, I think, for vitamin C. He said he was very interested in cancer and living systems. He said there's a paradox. He said, we know multiple things in the environment can elicit cancer. We've identified these: oncogenic viruses, chronic inflammation, carcinogens, intermittent hypoxia, rare germline mutations. He said, we don't understand the common pathophysiological mechanism by which any of those provocative agents would elicit dysregulated cell growth, which is cancer. When you talk about cancer, what is cancer? It's cell division out of control. It's dysregulated cell growth. This organelle determines when cells should divide and when they should not divide. It regulates the destiny of the cell. So what happens when this organelle becomes chronically impaired? It falls back on these ancient pathways, these pathways that existed before oxygen came into the environment, where all the cells were dysregulated in the growth because they didn't have this regulatory system, which is the mitochondria coming from a bacteria.
So let me play that back to you in a way, just so I make sure I understand.
I know it can be kind of deep.
Okay, so in this mitochondria here that I have in front of me, there is an ancient, because this came from the fusion of bacteria a long time ago. Yeah, that old bacteria,
about several billion years ago,
used to be very selfish and just think about itself. It used to grow on its own and didn't communicate with anybody, had its own way of growing and multiplying that was really not in cohesion with anything else. That is still in there somewhere. And although now, in its modern form, because there's been that fusion, it grows thinking about the wider organism, when it becomes damaged, it falls back on that old selfish way of growing. And that's kind of what can cause cancers. So if there's stresses on this, which could be all the carcinogenic things you described, then sometimes this falls back on that prehistoric, selfish way of growing, where it doesn't think about the wider organism.
It's close. In the sense of thinking, we don't know about that. Okay. We just know the consequences of what happens when it falls back on those ancient pathways. Now, here's the situation. People knew from Otto Warburg said cancer is a energy problem in the cell. Why would the cells start to ferment and produce a massive amount of fermentation waste product, which is lactic acid, even in the presence of oxygen, 100% oxygen? They're still fermenting. That shouldn't happen. When you and I hold our breath or have a heart attack, let me tell you something. This is really remarkable. And this is another piece of information that got us on this whole thing. When people have heart attacks, they stop breathing. The heart seizes. Okay, the bloodstream immediately fills with these fermentation waste products, which are lactic acid, and the other one, which we now know, is succinic acid. Okay, wow. These two waste products. Now, if you don't start breathing in a short period of time, you're going to be dead. Mm-hmm. And you die because the neurons in your brain cannot sustain this kind of fermentation energy for very long. As soon as you use the heart, you give them cardiac massage and whatever, the guy's heart starts beating again. The waste products of lactic acid and succinic acid go away. They disappear because you're breathing now. You don't need to ferment when you have oxygen in the environment. And the mitochondria can utilize the oxygen in the environment. Cancer cells, to Warburg said, it's the weirdest thing. These cancer cells continue to ferment even in 100% of oxygen. Why are they doing that? And he speculated that this organelle was damaged. Irreversible damage to the organelle happened in these cancer cells. He didn't have an electron microscope at the time. He didn't have the sophisticated tools that we have today. So he projected that all on biochemistry. At that time, he was a biochemist, and he said, you should not produce fermentation if you have oxygen should shut that off, and these guys should return to a normal metabolic homeostasis. And he said that's because there's something irreversibly damaged in this organelle. So a lot of people attacked him, and they said, oh, we don't have any evidence for that. Here's the beautiful thing. Some cancer cells continue to take in oxygen and make ATP. Therefore, Warburg must be wrong. We showed that. We showed the cancer cell takes in oxygen, but it's not making energy through ATP in any great amount. It's using it for ROS, these radicals that further damage and cause the DNA mutations that everybody is chasing. It's all downstream effects of damage.
So what does this mean in simple terms? Because I'm aware a lot of my views.
It means I say, okay, so I have dysregulated cell growth. That's ultimately what the problem we're dealing with. Mm-hmm. We can't control how these cells are dividing. We're throwing all kinds of crazy stuff at them, trying to poison or radiate, surgically remove them. We're trying to do everything to stop this dysregulated cell growth. Mm-hmm. So when we look at this organelle under the electron microscope, we find these cristae are often missing. You have what they call ghost mitochondria. You got the shell, but nothing inside. Or if they're inside, they're all deformed. So we know there's a foundational principle in biology. Structure determines function. If the structure is abnormal, the function will be abnormal. This is known to all biologists except oncologists. They don't seem to understand
What's
that.
an oncologist?
Those are the people who study cancer.
So what are you then?
I'm a biologist.
You're a biologist.
Yeah. I mean, when we know that if the organelle is damaged, you're not going to be able to produce energy efficiently by oxidative phosphorylation. Okay.
I think you're a bit further down the road, Thomas, than a lot of my viewers are. For me, hearing that there's this energy engine in my cells and there's trillions of them or billions of them, and they also communicate with each other. And when this becomes stressed or hurt or damaged because of lifestyle choices that I make, energy production and efficiency will change, then that could cause this to die or malfunction in some way. That is, for me, I go, I've got it.
Okay. That's a major step forward. And then we build upon that.
Yes.
Now we build upon that. Okay. So here's the situation. We've discussed cancer cells, all of them that we have ever looked at, have defects in the number, structure, and function of that organelle. Okay. I published that big paper where I spent over a year of my time going through the early electron microscopy literature. Warburg didn't have that opportunity because that technology was not there for him. So he speculated that based on the biochemistry. But I went back and I looked at these electron micrographs of mitochondria in various cancers, and they're all damaged. There's few of them. That cristae are gone. I worked with some of the best, like Arismendi Murillo, who was a world leader in beautiful electron microscopy of cancer cells, and you can see and all the damage under his magnificently beautiful. And we have a couple of papers. But let me tell you something else, Stephen. In the cytoplasm, these organelles are also in contact with other cellular membranes, like the endoplasmic reticulum. There's a lot of what we call organelles inside a cell. You have the nucleus, you have the mitochondrion, you have lysosomes. There's intimate contacts between some of the other membranes, mitochondrial associated membranes we see. And they're also abnormal when you look at them under the electron microscope.
The ones it's talking to are abnormal.
Yeah, the ones that you can see. The mitochondria are abnormal and the membranes that contact them are abnormal.
Okay.
And that intimate contact, I'll get into the calcium signaling a little bit later, which controls the destiny of the cell. Why the cell is growing out of control? Well, first of all, it's fermenting. Okay, that means it's getting energy from sources other than oxidative phosphorylation. You can take a cancer cell and treat it with cyanide or azide, or in absence of, and it's still living. It's still growing because it's not using
the oxygen pathway.
They're not using the oxygen, falling back on oxygen independent mechanisms, which are called fermentation.
So I understand that to be that there's a malfunction in the mitochondria, which means that it no longer uses its oxygen pathway necessarily.
As sufficiently as it should. There's always some residual level.
And it finds another way to make the energy, which is how it survives.
Yes.
And then it stops communicating with the rest of the cell.
Well, it actually communicates with the nucleus to open up the floodgates to bring in the fuels that drive this fermentation energy.
It gets more and more greedy.
It has to, because you're taking an organelle that produces energy highly efficiently, okay, like 34 to 36 ATPs.
With oxygen.
With oxygen. And now you're trying to replace that with fuels that give you 2 moles of energy.
So it's inefficient.
And then you get 2 out of so you're getting 4. So you have to take in order to make up the efficiency, you must have a tremendous logistic. You must have tremendous supply of the fuels that will give us energy.
Which are?
Glucose, the sugar.
Yeah.
And the amino acid glutamine.
Okay.
Our bodies are loaded with glutamine. That's the most abundant amino acid in our bloodstream. And evolution provided that for us, because if we stop breathing, we use that fuel to keep the cells alive. Our gut is controlled by glutamine. Our immune system uses glutamine.
What is glutamine?
It's an amino acid.
Which we make from food.
Yeah, which we can make from food, true. Or they call essential and non-essential amino acids. Essentials are from that we must eat. We must have certain foods that provide these. Glutamine is considered a non-essential amino acid. It's an essential amino acid, biochemically called non-essential, because we can make it from sugar. But basically, it's the most abundant amino acid in our body.
I think I get it now.
Okay, feed me back. Tell me. Let me test you.
There should be a button just down below here. And if it says subscribed, you're already subscribed. If it says subscriber, that means you're not yet. And if you're not subscribed, please could you do us a favor and hit that button. It helps the show more than you know. And according to the algorithm, you're someone that watches our show, but you haven't yet hit that button. Thank you so much. When we fall, the mitochondria falls to that prehistoric pathway where it starts making energy in a really, really inefficient way without using oxygen in the same way. Yep. It starts relying now on glucose and glutamine to produce that energy, which is a much less efficient source of energy. So it gets a bit more greedy and it needs much more to make the same amount of ATP, which is energy. Mm-hmm. So a cancer cell can be very, very greedy. It's not responding to oxygen in the same way. And they're a bit more selfish. They start sort of multiplying without thought of the broader organism.
Yes.
So the question I have is, how does this happen? Malfunctioning of the mitochondria. What is it that I've done? You know what else this question
okay,
well, it's because children can get these cancers.
Right.
Or a 90-year-old can get the cancer. Yeah. And it doesn't appear to me that a 3-year-old has necessarily made life choices yet that can impact.
But as I said, when you consider forever chemicals that are in the environment, things that we use in our technologically advanced societies can get through the placental wall and get these into these children's organs. You have to put it together. Usually it's a constellation of things, because don't forget, I said carcinogens. Some of these carcinogens are fat soluble. Some of these carcinogens can get in and damage those during early stages of development. People always say, how do you explain brain cancer in a 6-month-old baby? Okay, he wasn't smoking and drinking and partying all the time, but the mitochondria and a population of cells in his brain became damaged, and that damage then led to this dysregulated cell growth, because the organelle controls when cells should divide and not divide. And then where's the energy coming from? How do you take a highly sophisticated piece of an organelle and making energy so incredibly efficient? And now we're using energy. So this organelle signals to the nucleus, called mitochondrial stress response, retrograde signaling. The nucleus acts as a respondent to what this organelle wants. So the nucleus then oncogenes, that you've heard a lot about, they open the floodgates to bring in the glucose and glutamine that allow the cell to grow in a dysregulated way. So as you said, they go back to these ancient fermentation pathways where there was no regulation, because this organelle was not part of the problem. So not part of the situation, the regulation.
So is there, what are the lifestyle factors that are causing this drastically increasing the probability of this happening?
It's not necessarily what the person did in all cases. It's what the person was exposed to that could have elicited this. That's the oncogenic paradox. So we have shown that inflammation produces these cytokines. When you have an inflammatory inflammation, they damage the ability of this organelle to make energy efficiently. Chronic inflammation is known to be a risk factor for cancer. Chronic inflammation. Intermittent hypoxia. Carcinogens.
What's intermittent hypoxia?
It's like the sleep apnea, that kind of thing.
Oh, okay.
Warburg had clearly shown that intermittent hypoxia on cells would damage the efficiency of oxidative phosphorylation, leading to a compensatory fermentation. So you have to compensate, because let me tell you, if you don't compensate, you're dead. the cell dies.
When we look at the populations around the world that have the most prevalence of cancer, it doesn't appear to be some of the countries like Niger, Gambia, Nepal consistently rank at the very bottom for cancer incidence. Conversely, high-income countries like Australia, New Zealand, and the United States have the highest rates of cancer. Why is that?
It's because of our technology. We are still Paleolithic man, and our biology has allowed us to store energy efficiently because of times of famine. We are now in a new environment where we have massive amounts of highly processed carbohydrates, inactivity, emotional stress. We have poor sleep habits. You pile all those together with the exposure to carcinogens and whatever, and you chronically damage this organelle. And in some organs, you can get breast cancer, if it's a lung, if it's whatever it is, that organelle becomes chronically damaged in some populations of cells in a particular organ. And you can elicit dysregulated cell growth as the result of that. What I find is in the paper that we have with the chart, if you can keep your mitochondria healthy, because don't forget, Paleolithic man, our ancestors from 500,000 years ago, or modern men, like you said, in these countries, living according to traditional ways with minimal interference from modern diet and lifestyle issues, have lower amount of cancer in general. And this is what Albert Schweitzer found, the famous humanitarian physician. He was specifically looking for cancer in African tribes. And he said, remarkably, it's extremely low. What are these guys doing where Western society has a lot of cancer and these Africans have living according to the traditional ways? So they have a lot of exercise. They're eating all organic foods. They're not under the same kind of stress or exposure to chemicals that modern societies have. And you find out you have very low cancer. Like, for example, dogs are all evolved from the wolf. Wolves in the wild rarely have cancer. The domestic dog, cancer is the number one killer of the domestic dog. What is the dog doing that the wolf? The wolf is out running around eating natural foods. The dog is in an apartment somewhere. It's a dog walker once a day, right? And the next thing, the dog is obese and full of cancer. So it all comes down to what you do to maintain the health and vitality of the mitochondria that reduce the risk. And that's what this chart I'll show you has. Reduce the risk of damaging this chronically. Okay, so that can explain in large part why modern societies are struggling with chronic diseases, not only cancer. We have type 2 diabetes, we have obesity, we have high blood pressure, we have a whole, even neuropsychiatric problems. If you can protect and keep this organelle healthy, you reduce risk. Now, people say, well, cancer has to be genetic because we have inherited genes that put us at higher risk, like the BRCA1 for breast cancer and the LIFRA-many for a variety of other cancers. Our paper in this pile, done by Bob Kaplan, he went through and looked at all the genetic risk factors.
Which paper is it?
It's one of the ones published in Oncology. But none of these mutations are 100% penetrant, meaning that they're secondary risk factors. A primary risk factor would be every time that mutation is there, 100% of the people, because I work in Tay-Sachs disease, I work in more areas of metabolism. Those mutations are 100% responsible for that condition. There's no gene mutation that's 100% penetrant. You have that gene, you're going to 100% get cancer. Most of them are what they call incompletely penetrant.
So what does that tell us about the nature of...
Well, and then we went back. What Bob did is he went back and he looked at what every one of those gene mutations in some way disturbs the efficiency of oxidative phosphorylation in that organelle.
In the mitochondria.
In the mitochondria. We looked at, we have all the evidence, all the risk factor, all the genetic, just the front page there. I was going to show you the hard data, but you got it. So anyway, all of them damage the efficiency of energy through this organelle. So that's like carcinogens. That's like viral infections. The viruses that hepatoma and papilloma, their products will go in here and damage it, or they will replicate inside this organelle, screwing up the efficiency, causing a compensatory fermentation, causing the dysregulated cell growth through abnormal calcium signaling, causing cells to no longer be responsive to their neighbors. Is that clear? if I can make you understand this, we can make everybody understand this.
Yeah. So the reason I spend a lot of time asking why and asking for clarification is because when I do this show, I then go out into the real world and I meet the people that listen.
Yeah.
And one of the groups of people that listens are young offenders. And when I went and visited them, I pointed at the episodes that I thought would help them.
Yeah.
And one of the young offenders said to me, I can't listen to that episode because the words you used were too big. And I remember thinking to myself,
okay, I get that all the time, but guess what? Now we have AI, and you can take this statements and put it into AI right on and it'll dumb it down for you. Mm-hmm. So that's a tool that we previously did not have. Mm-hmm. So I use that all the time. I said, when you hear me speak like this, don't think I'm arrogant. Don't think I'm just, these are the terms that we use when you're part of the academy.
Yeah.
Okay. But we can take those terms now, and AI can do a wonderful job in synthesizing. Oh yeah, I know what he's talking about now. But without that tool, then it becomes like you said, well, I can't understand anything.